And this is where we left off. We'll move on to the next slide. This slide gives you the beginning of an overview of how urine is produced. It's produced in three basic steps. Filtration, which this is filtration of the blood entering the glomerulus. So blood enters the nephron, and the first part of the nephron is the glomerulus. It is then filtered by size. Those things that are small enough will fall through. Those things that are too big will stay in the blood. Normally no cells, no blood cells, because they're too big, or large proteins are going to enter the filtrate. So what you have are small toxins, ions, water, other glucose even can can slip through, will slip through openings or slits, little holes that are in the glomerulus, into the lumen, or the negative space. We call it the space in the glomerular capsule, and then start its journey. That liquid will start its journey through the rest of the nephron system. In the journey through the rest of the nephron system, there will be a time when reabsorption takes place. Anything that the body can use, so nutrients, water, ions, are reclaimed and returned to the blood. These, remember, were too small. They were small enough to actually pour through the holes in the filtration process. And then secretion. Anything that's undesirable are actively removed from what is are removed, or they actually stay in the tube system, and it is secreted out in a fluid mixture which we call urine. So three basic portions that take place in urine production: filtration, reabsorption, and secretion. And we'll spend basically the rest of the teaching times talking about breaking that down in a little bit more detail. Two types of nephrons you have, depending on where they're found. You have cortical nephrons. They are the most abundant type. As a matter of fact, about 85% of your nephrons are cortical, and they have they are so named because of where they're found. They're found in the cortex. They have the nephron has a design where you have a glomerulus, and then you have a tube system that has a long loop on it called a loop of Henle. And with cortical nephrons, the loop of Henle doesn't extend very far in the medullary portion of the renal pyramid. But the other type, which is the juxtamedullary, are found at the space between the cortex and the medullary, and their loop of Henle hangs very deeply into the medulla of the renal pyramid. The juxtamedullary nephrons are able to create very concentrated urine because of that extended loop of Henle. Loop of Henle is that loop that's hanging down. Back to the renal corpuscle. Remember, we're going to go into those three stages of urine production in greater detail. So we're looking at how does this filtration take place. The renal corpuscle, the actual definition of a renal corpuscle is the glomerulus and the capsule together. The glomerulus is the extension of that that renal artery. Remember, is going to divide into smaller and smaller units and eventually become the afferent arteriole. That afferent arteriole is going to turn into little tufts of capillary beds, which we call the glomerulus. That is housed in a structure called the capsule, which is made up of epithelial cells, connective tissue, and a space in between. And so the blood pours in. Those things that are able to fall through slits, because the capillaries in the glomerulus are fenestrated. Fenestrated capillaries have spaces between their cells, their endothelium. Endothelial cells make up the smallest one cell version of capillaries, and the junctions between each cells are not particularly tight, so you have what's known as fenestration, and things can fall through. Then you actually have specialized cells around those fenestrated capillaries. We call them podocytes, and they have slits within them. We have a nice image coming up which might help you out with that. The only other thing I want to show you now, just to let you know a little bit ahead, you have afferent arteriole, efferent arteriole, and then you also have this juxtaglomerular cells, and sometimes it's called the juxtaglomerular complex, that we will refer to, that has specialized function with regard to what the kidney does for us. And then here's another image of those podocytes, where you have they actually surround the tufts of the capillary beds, and you're seeing a little bit of the fenestrations. It looks like holes, but actually it's just where certain cells aren't fitting together as well, but on purpose. They're fenestrated capillary beds. The products from the blood will pour out, water, ions, etc., pour through there, and then also pour through the slits, and then into the space between the glomerulus and the capsule, so that capsular space. So you have on the right, you have capillaries and the space in between individual cells. That's the fenestration. Basement membrane, the only reason to think about that is basement membranes tend to have a charge. You have charged proteins on there, and if you have something that has a charge, it also is going to act as a deterrent of something passing through. But if in fact it manages to make it out of the capillary bed, past the basement membrane, because of a charge or a lack of charge, or solubility, then it's going to fit through the slits into the capsular space. The other thing about this area, you have a cell called a mesangial cell. And it is, and what you're seeing it here, this doesn't come from your textbook, but I thought you have one in your textbook. It's not quite as detailed. Just an FYI is, so you see the cell is between the capillaries. You have two capillaries there. You see the cell there, and what happens is, based on regulation, a chemical called angiotensin II or ADH or histamine can act on that cell. That cell's cytoskeleton is made up of contractile proteins that can cause that cell to contract or expand and actually affect the size of the capillaries. So the capillaries are a single cell, so you don't have smooth muscle in there, but you do have this particular cell that actually can control the flow of blood going in or out of this particular glomerulus. And then connecting to the renal corpuscle, remember the corpuscles, there's two together, is a tube system that then is responsible for the reabsorption and secretion. All filtration takes place in the glomerulus, or the glomerular capsule, or the renal corpuscle. I tend to use those three terms interchangeably. Please understand that I'm referring to that area, I guess, when I'm using them interchangeably, I guess. I guess I should correct that in myself. And then the first portion, so the portion that's closest to the renal corpuscle, is called the proximal convoluted tubule, and that's a mouthful, so we abbreviate that as the PCT. And it's surrounded by capillary beds, those paratubular capillaries that we talked about in a previous slide, that are there to catch and absorb things that reabsorb things that made it through the glomerular capsule and but are still needed by the body, like water and solutes. And then from the PCT, that long loop happens, which we call the loop of Henle, and that term does get capitalized because it's named after a person. You have the descending limb and the ascending limb, and each of these has its own unique function with regard to urine production, so we name them individually because they have an individual function. The descending and ascending limb then feeds into the distal convoluted tubule, so called because it's the further away from the glomerular capsule. Distal convoluted tubule then connects to the connecting tube. Connecting tube will lead into the calyces. You have actually a papillary duct that then leads into the minor calyces, into the major calyces, into the ureter, so that the urine then can move on out. Blood vessels in the, particularly the juxtamedullary nephrons, is called the vasa recta, because it's a unique type of blood organization that can act on the what then is the filtrate in a much more intense way. Collecting tubes, their job is to collect urine from several nephrons. So what we've done is we've moved on out of the nephron into this collecting tube. I kind of talked about it in the last one. That then is going to, so it functions for several nephrons, empties into papillary ducts. You see the term there, P-A-P-I-L-L-A-R-Y, ducts, then empties into minor calyces. Minor calyces again become major, and then on out the body. The collecting tubes are not, have the ability to shift their permeability to water. So if water is very valuable for the body, so you're dehydrated, they become less permeable to water. If you don't need the water, so you're appropriately hydrated or maybe too hydrated, they can become more permeable to water. This happens because aquaporins, which are water channels, can be inserted and taken out. The filtration with regard to what comes out of the glomerulus is mostly water and solutes. And then it's a non-selective process. So it is passive and just based on, I guess, non-selective except for it's based on size. Blood cells usually cannot pass out of these capillaries. Things that control glomerular filtration rate or just control the filtration, it has to do with pressure. So it's a passive process, and we know passive processes, things go from high concentration to lower concentration. So as the blood is coming in, this is arterial blood. Arterial blood, we know by definition, even if you get into these capillary beds, is going to be under pressure. So it comes in and it is pushing out at about 50 milligrams of mercury. And you do have, you eventually have pressure of the filtrate that's in the space. There'll be some that's left over, but it is only going to be minuscule. So there actually is a positive pressure of the blood and the things that are coming out of the blood toward the capsule space of 10 milligrams of mercury. So things are going to move out just based on hydrostatic pressure. So the first definition you have there is hydrostatic pressure. And we'll pick this up on the next one.